Welcome to Talk Tutors, where I'll be showing you step-by-step guide on how to ace exam questions. Get a pen and a paper and listen. So figure 9 shows an area of woodland with some small plants growing in the ground between the trees. Okay, so you can see the small plants. The leaves of the small plants are green. Describe how these plants make glucose. Describe how the plants make glucose for 2 marks. So I need 2 points for 1 mark each. And describe, so I need to state facts. Okay, so I know already that plants need CO2 plus water, then plus light from the sun to make glucose and release oxygen. So this is what I know. So how do I put that in writing? For plants to absorb light, they need to have chlorophyll. Okay, so I'll have to write down plants have chlorophyll inside chloroplast to absorb sunlight. That's 1 point. My second point would be the CO2 taken in through the leaf reacts with water to make glucose through photosynthesis. Okay, that's my 2 marks. Figure 15 shows a diagram of some equipment that can be used to investigate the rate of photosynthesis. You can see a light source, conduit, water, bubbles of oxygen. Devise a plan to investigate the effect of light intensity on the rate of photosynthesis. Include variables you would need to control. For 6 marks, and it says devise. So devise means I need to write a method for the experiment. So I need 6 different points. Now get me a mark each. From my first point, I saw a light source. I have a light source. So I'll bring back the diagram. I have my light source and I have my conduit. And I know that for oxygen to be produced, plants need light, and they need water, and they need CO2. So for me to investigate the rate of the effect of light intensity, I need to be able to move the light source either closer and count how many oxygen is produced, how many bubbles are produced, or I move it further away to see if I'm producing more or less oxygen bubbles. So what would I write first? My step 1, place the light source at a known distance away from the conduit. If we're investigating light intensity, we either increase light intensity or decrease it and see the effect by counting the bubbles. So my second point would be, so when I've placed the light source at a known distance, I need to count the number of bubbles in a given time. Okay, example could be 1 minute. Example of a known distance could be 10 centimeters. Doesn't matter, whatever you want. Now after I've done this, I need to now change my distance. So my third point would be, repeat the experiment at a different distance between the light source and the conduit. So you can move it closer or you can move it further away and count the bubbles to see the effect. Do you have more bubbles or less bubbles? So when I've done that, my fourth point would be, I can keep the amount of conduit should be kept the same. Okay. 5, when you place the light source closer to the conduit, remember light source is it will transfer heat onto the conduit, and you don't want that. Okay, so what do we need to do? We place a sheet of glass. You place a sheet of glass between the light source and the conduit to stop it from heating up. Okay, so this in point 2, we have count the number of bubbles. That's 1 mark in a given time, gets us 2 marks. So in total we have 6 points. Algae are green plants. Figure 10 shows the number of algae in a lake in the UK during 1 year. So January to December. Okay, I can see that from January to around, that's the peak. So somewhere mid-June, July. So here it is increasing. So there's a peak and then it begins to decrease. So before I even read the question, this is what I can see from the graph. Okay, number of algae would increase from January to around mid-June, peak around mid-June, and then decrease from July to December. Okay, that's what I can see before I look at the question. So the question is saying, explain the changes in the number of algae in the lake from February to June. The changes in number of algae from February to June. Bring back the graph. So we can see February to June, there is an increase in number of algae. So our first point would be the number of algae increases. Okay, that's our first point. Now, explain. It says explain means we need to give reason. So what is the reason for our point? This is because from February to June, the temperature is increasing because we're getting towards summertime. So because of increase in temperature, or we can say light intensity, because it's getting hotter, remember, and there's more sun. So if there's more light intensity, what does that mean? That means that the plants will absorb light and make more food and grow more. Okay, so more photosynthesis will occurs, and more algae plants grow. Okay, so we have our this is our point, and we have our reasons for 3 marks. Figure 9 shows an area of woodland with some small plants growing in the ground between the trees. So similar to the first question, a scientist recorded the mean light intensity. Mean is average, so the average light intensity and the mean number of small plants per square meter for 6 25 meter square areas of the woodland. So A, B, C, D, E, F. So that's the 6 of them. Light intensity, the average light intensity is decreasing. Okay, and the mean number of small plants is also decreasing. So as the light intensity decreases, the number of small plants are also decreasing from the table. Okay, so the question says, explain the effects of light intensity on the number of small plants for 2 marks. And it says explain, so I need 1 point and 1 reason. To explain, need to give a reason. Okay, so I know that as the light intensity is decreasing, the number of small plants are decreasing. So write it down. Okay, so why is that? This is because the small plants will not be able to carry out photosynthesis enough. Remember, they're small and they're covered around big trees, so less light absorbed means that they will not be able to photosynthesize enough. Okay, so that's our reason. State one variable the scientists should have controlled to make the light intensity measurements, to make sure the light intensity measurements could be compared. What could we keep the same while carrying out this experiment? We could carry out the experiment at the same time of the day. Okay, that could be a control variable. We can also say same person needs to take the measurement. So same person takes the measurement. We can also say using the same equipment. We can use the same equipment, but these two can do, because this is just 1 mark. Explain the effects of increasing air temperature on the rate of transpiration. If we are increasing the air temperature, that means it's getting hotter. Okay, that means it's getting hotter. So if it's getting hotter, and I know transpiration is loss of water, so if it's getting hotter, that means water will be lost faster because of evaporation. So for 2 marks, it says explain, so that means give a reason. So this is what I know. If you increase air temperature, that means it's getting hotter. Transpiration means loss of water. So I write down the transpiration rate will increase because water molecules have more energy, so will move faster and become gas. Okay, so when they become gas, they will evaporate. Figure 10 shows the leaves and flowers of water lily. So you can see the leaves and flowers. Water lilies have stomata on the upper side of the leaves. Explain why water lilies have no stomata on the lower surface of the leaf. Now, if we bring back the image, so they have stomata on the upper side, but not on the lower side. And if they are on water, like it says, flowers and leaves on water. So if they are on water, the side underneath, so the lower side, is not really coming in contact with the air. Okay, so there must be a reason for that. How is it adapted to do that? So we say the stomata, for instance, is where there's gas exchange. So gas exchange happens at the stomata, and there's a reason why if the leaves are facing the upper surface, that means that the leaves are coming in contact with CO2. Okay, they absorb CO2 and then they remove O2. So that's the gas exchange. Okay, gas exchange happening. But underneath the surface, so on the lower surface, there's no gas inside the water, so there is no gas exchange occurring on the lower side because it is facing the water. So what would we write down for our 2 marks? And it says explain, so we need to give a reason for our point. So our point would be lower surface of leaves is not in contact with air, so no gas exchange occurs, therefore no need to have stomata. Scientists can measure how much water is lost by the leaves of plant. What is the movement of water molecules? So when he talks about water molecules, that is directly talking about osmosis. So I don't really need to look at the question. The answer will be osmosis. Diffusion will be for gases. Active transport, nutrients, normally in the roots of plants. What structure transports water through the stem of plant? Transports water. Okay, that would be so option A says guard cell, B phloem. So I know that phloem and xylem are the stems of plant, but one of them transport water, and that is the xylem. Figure 10 shows the leaves and flowers of water lily plants. So you can see, similar to the previous question, the white petals of the water lily flowers cannot photosynthesize. Which structure in the leaf cells is the site of photosynthesis? The site of photosynthesis. So which site absorbs sunlight? That would be the chloroplast. Glucose is made by photosynthesis. Glucose is converted to another sugar to be transported in the plant. What is the name of this sugar? So it's not glycerol, it's not ribose, it's not starch, so it will be sucrose. That's for our 1 mark. Describe how this sugar is transported from the leaves to the flowers of the water lily. Remember, xylem transports water. So if it's nutrient like sugar, it will be transported by the phloem. So it says describe how this sugar is transported. For 2 marks, our first answer will be in the phloem. Second point, how is it transported in the phloem? The sugar is dissolved in water. Okay, our point 3, the sugar is transported using active transport. Okay, for 2 marks, we've written 3 points. A scientist investigated how the flow of air affected the rate of transpiration. Transpiration, like we know, is loss of water. Okay, a fan was used to change a fan was used to change the flow of air. The volume of water taken up by the plant was measured. So volume of water taken up is measured, and we can see that on the graph. So this graph is for when the fan is switched on after 3 minutes. This is fan not switched on. The volume of water taken up by plant, millimeters cube. So we can see more volume of water is taken up by the plant when the plant is switched on than when the plant is not switched on. Why is that? So when you put a fan, so let's picture that. When you put a fan blowing air on the plant, okay, if the fan is moving faster, or if the fan is on, water droplets are going to be lost. Okay, so imagine there's water on the leaf of plant and a fan is blowing air, the water droplets are going to be lost. And if a plant loses water, it needs to take up more water. So the plant's roots need to take up more water. Okay, so that makes sense why the volume of water taken up by the plant will increase when the fan is on. Because when it's on, water is lost from the leaves. That means it needs to take up more water. If not, the plant is going to dry out, and we don't want that. So, but when the fan is not switched on, the volume is a bit less, or a lot less. Okay, so what are we going to write down? Explain why switching on the fan caused a change in the volume of water taken up by the plant. So for 3 marks, it says explain. So I need a point and 2 reasons for 1 mark each. So explain means give reason. So I start with my reason. The fan turned on causes water to be removed from the leaves. Okay, so that's one. Water is going to be removed from the leaves. So this increases the rate of transpiration. Okay, so that means if the fan is on, there's an increased rate of water lost from the leaves. This causes the plant roots to absorb or take in more water. Okay, give 1 reason why the volume of water taken up by the plant was also measured when the fan was not switched on. Okay, why do we need to measure that? So we can have a data to compare our results to. Okay, so we have data to compare the effect of the fan to. Okay, so it's just a control variable that gives us 1 mark. Calculate the rate of water uptake after 8 minutes. Calculate the rate of water uptake from 8 minutes to 10 minutes when the fan was switched on. So at 8 minutes, we need to find the water uptake. And at 10 minutes, we need to find the water uptake. So let's bring back the graph. So bring this graph back so we can see at 8 minutes, we get our ruler. 8 minutes and 10 minutes. So 8 minutes will be here. So the value here would be, so this is 50, 51, 52. So that would be 50, 52 for 8 minutes. And 10 minutes would be, say, 65, 66, 67, 68. So 10 minutes would be 68. So at 8 minutes, that will be 52 millimeters cube. At 10 minutes, that's 68. So the rate of water uptake would be 68 minus 52. The time taken, difference of time taken would be 2 minutes. So 10 minus, that would give us 8 millimeters cube per minute. So here we write 8. A scientist measured the rate of water lost, that's transpiration, from a plant shoot using a potometer. Figure 8 shows the equipment used in the experiment. So leafy twig, reservoir, bubble, capillary, beaker containing water. Okay. The volume of water lost from the plant can be calculated by measuring the distance a bubble moves along the capillary tubing. Okay. Distance a bubble moves along the capillary tubing. All right. Calculate the rate of water lost from the plant in millimeters cube per second. If the volume of water lost was 12 millimeters cube in 10 minutes. So if it wants answers in per second, but it's giving us time in minutes, and this is a 3-mark question that says calculate, so we need to show working out. So 10 minutes would be, that would be 10 times 60, so that would be 600 seconds, because it wants answers in per second. Okay, now water lost is 12 millimeters cube per 10 minutes, so that would be 12 millimeters cube in 600 seconds. How many would there be in 1 second? So what do? 12 divided by 600, that gives us 0.02 millimeters cube per second. Explain how the water lost would change if the plant only had 1 leaf. So if there was only 1 leaf, how would the water loss change? So now let's visualize this. If we have a plant that has multiple leaves and a plant that has only 1 leaf, which of them do you think would lose water easily? It would be this one. So because there's more leaf, and the more leaf means there's more contact with the air. This only has only 1 leaf. So the rate of water lost in this plant with 1 leaf is going to be less. So explain, we need to give a reason. So our first point would be the rate of water lost. Water lost is transpiration. So the rate of transpiration would reduce if it had only 1 leaf. This is because of less evaporation of water vapor from the leaf, not leaves, to the air. Okay. So the rate of transpiration would reduce if it had only 1 leaf because the less evaporation of water vapor from the leaf to the air. That gives us 2 marks. The scientist wants to extend the investigation by considering other factors. that affect transpiration rate. What other factor would affect transpiration? Remember, this means water loss. So for 2 marks, what could we say? If it's not the temperature of the surrounding air, it could be the air speed. So if the air is moving too fast, there will be water vapor lost from the leaf. Remember, when you turn on the fan, it increases the rate of water loss. What else could affect transpiration rate? It would be if the place is humid. So I would say humidity. Another factor could be light intensity. Okay, but it's 2 marks, so you can give any of these 3 points. The rate of photosynthesis can be measured using the equipment shown in figure 3. So we can see light source, a lamp, conduit, bubbles of gas coming out that we have to count, water plus sodium hydrogen carbonate. Okay, a student investigated the effects of carbon dioxide on the rate of photosynthesis. Sodium hydrogen carbonate provides the conduit with CO2. Name 2 variables that the students will need to keep constant in this investigation. So we need to keep the temperature the same. Temperature needs to be kept the same because if it gets hotter, that will make a difference. The mass of the conduit should be kept the same. What else could be kept the same? The volume of the solution should be kept the same. What else could we keep the same? Distance of lamp to the conduit. Distance of lamp to conduit should be kept the same because we are investigating the effects of CO2, not light intensity. Remember, not light intensity. So we have to keep the light intensity the same. So again, we need to keep the light intensity the same. So that gives us our 2 marks. Which will be the best piece of equipment for measuring volume of gas collected? So we covered this in paper 1 of our chemistry, when we talked about the rate of reaction and if a gas is produced. A gas syringe is always used to collect the gas. So that looks like this. So this equipment is a gas syringe. We use that to collect gas. The students added different volumes of sodium hydrogen carbonate solution to the water and measured the volume of gas collected in 1 hour. Okay, so we have our tables. Volume 0, 5, 10, 15, 20, 25, 30. So we can see as the volume is increasing, the volume of gas collected is also increasing, but up until this volume. So this stays the same. So this increases and this stays the same. So there's no change here. Okay. So from 20 centimeters cube up to 30 centimeters cube, there was no difference. Okay. Question. Describe the effect increasing the volume of sodium hydrogen carbonate solution on the volume of gas collected. Describe means state fact. And for 2 marks, we just need 2 facts for 1 mark each. So what fact do we know? As the volume of solution increases, the volume of gas collected would increase. So we have to write down, as the volume of sodium hydrogen carbonate solution increases, the volume of gas collected increases. That's our first point. Our second point. So if you add more than 20 centimeters cube of solution, there is not a change in the volume of gas collected. That is what we can see from this table. So if you add more than 20 centimeters cube, it still remains at the same 6. So there's no more change. Figure 5 shows nettle leaves taken from plants on 2 sides of a hill. So this is bigger or larger and this is smaller. Okay, shaded side of the hill. So that means no sun. The side not facing the sun. Okay, the sunny side of the hill facing sun. So why is the side facing the sun smaller than the side not facing the sun? No sun facing. Why is that? So explain the difference in surface area of the leaves on the shaded and sunny side of the hill. For 2 marks, it says explain. So we need the points for 1 mark and our reason for 1 mark. Explain means to give reason. Okay, so what will be our point? So we can say the shaded side leaves have a larger surface area. That we can see from the diagram because remember, it does not get enough sun, so it needs a larger surface area to absorb light. Okay, to absorb any light that it can take because it has less chance of getting enough sunlight. So because it needs increased light absorption for photosynthesis, so that's why it has a larger surface area. Figure 9 shows xylem and phloem from the stem of a plant. So xylem transports water, phloem transports minerals, okay, sugar. Living cells in phloem use energy to transport sucrose, that's sugar. So sucrose is sugar. Which organelle releases energy in living cells? And that would be the mitochondria. Mitochondria is where energy is released from respiration. Describe 2 features of the structure of a xylem vessel that can be seen in figure 9. So you can see from this image, the xylem has thicker walls than the phloem, and it's more hollow. It's a continuous hollow tube than the phloem. The phloem has these barriers. So we write it down. Xylem has thicker walls. Also, it has continuous hollow tube and no barriers. Okay, so that's 1 mark and 1 mark. For 2 marks, we have stated the fact. Complete the word equation for photosynthesis. So carbon dioxide plus water, we have light, then we have glucose plus oxygen. That gets us 1 mark. Describe the vessels that transport water up the stem. So water, that would be xylem. So we write down for 2 marks and it says describe, so we need to state facts. Facts would be hollow xylem. Our second fact is it has to be thick walls. Okay, that's a fact about xylem. Third fact will be it has to be lignified. That means dead. Okay, so those are our marks. If you have found this video useful, please like and leave a comment. Subscribe and share with your friends. The aim of the video is to help you. Until next time.